Good evening everyone. I welcome you to this new lecture video. So basically today we are going to look at enzymology in biochemistry. We are going to look at enzymology. So enzymology, as we all know, it is just basically the study of the same enzymes that we are going to talk about. So something that is very interesting is about enzymes is that they are involved in almost all body processes and mechanisms that takes inside there, because these enzymes are very, very important. You're going to find that there are enzymes that helps in digestion, there are enzymes that helps in different body metabolic reactions. So this shows that enzymes are actually very, very important, because even in DNA transcription, translation, those things, you're going to find that enzymes are very, very important. They play a major role in those processes. So we're going to start to look at enzymes, what the enzyme is, and different classification of enzymes. We are also going to look at the types of inhibition of the same enzymes. Also, we are going to look at the regulation of the same enzymes and enzyme kinetics, how these enzymes operate, and so on and so forth. So stay tuned. Where you have a question, feel free, and from there we are going to move. So let's start by defining what an enzyme is. So an enzyme, basically, this is a familiar concept, rather term now that we all know. So enzyme is just a biological catalyst that tends to speed up the rate of chemical reaction, or it is a biological molecule, which is typically a protein that speeds up the rate of chemical reaction, and also it lowers the activation energy of that particular reaction that it is carrying out. So we know that an enzyme has got a mechanism. So it has got just specifically a mechanism that it operates under. So we know that if you are having an enzyme, let's say here, this is the enzyme that we are having. This enzyme has got parts. So it is going to have the allosteric part, also the active site. So at this moment, how an enzyme operates is that on this active site, the active site has got a specific shape for a specific substrate that binds on it. So when a substrate comes, let's say this is the substrate that we are having, binds to this enzyme, it is going to form what we call enzyme substrate complex. So when that enzyme substrate complex is formed, this enzyme will now carry out the process of now converting this substrate molecule into products. So this is just the mechanism that an enzyme follows. When a substrate binds to its active site, it will convert now that substrate into products that are going to be formed. So basically this is the mechanism for the enzymes. So now these enzymes, they differ, because we know that there are enzymes that specifically just for that are involved in DNA transcription, translation, those that are involved in the breaking down of molecules, and so on and so forth. But this mechanism is that they have got the active site, and that active site has got its specific substrate that binds onto it. So one thing that you have to know, that the enzymes, they speed up the rate of chemical reaction, and most enzymes are protein in nature. So enzymes are protein in nature, most of them, but it is not every enzyme that is protein in nature, because we have enzymes like RNA polymerase and other enzymes that are not proteins. So it's not every enzyme that is protein in nature, but most of the enzymes are protein in nature. That's why they're just classified as enzymes are protein in nature. That is because the majority of the enzymes that are known and that are involved in most of the body processes are protein in nature. So you're going to find that enzymes, they are classified according to the type of reactions that they perform. So we have got different types of enzyme classification. So this enzyme, they're going to be put in a specific class according to the type of reaction that they perform. So the first class that we're going to look at is an enzyme class that is known as oxidoreductase. So this oxidoreductase enzyme, these are enzymes that are involved in a reaction that is an oxidation reduction reaction. So they are involved in the transfer of an electron from one molecule to another. that's the function of these oxidoreductase enzymes. So you're going to find that if you are having a molecule which is molecule A in a reaction is reacting with molecule B to produce another molecules that are going to be products. If this is an oxidative reductive reaction, what will happen is that this molecule here is going to, an enzyme that is an oxidoreductase enzyme that falls under this class will get one electron from this molecule and it will transfer it to the other molecule. So meaning that one molecule is going to be reduced and another molecule is going to be oxidized. That is where the concept of oxidoreductase enzyme classification is coming from. So we have examples of these oxidoreductase enzymes such as dehydrogenases, oxidases, reductases. These are some of the enzymes that falls under oxidoreductase classes because they are involved in the transfer of electrons between one molecule and another, and hence they end up forming a molecule that is going to be oxidized and a molecule that is going to be reduced. Then we also have enzymes that are known as, a class of enzyme that is known as transferase enzyme. Transferase enzyme. So these transferase enzymes are involved in the transfer or in the reaction that involves the transfer of a functional group from one molecule to another. So we are going to find that when a functional group is moved from one molecule to another, there is going to be a formation of a product. So a functional group is basically an atom that gives a particular molecule its characteristic chemical properties. Yeah, and that enables that molecule to undergo the same reaction that it is involved in. So we're going to find that functional groups like the methyl groups, the phosphoryl groups, they are going to be certain types of groups that will be transferred by enzymes that falls under this class, which are transferase enzymes. Just from the term transfer, you know that these enzymes, they will be involved in the transferring of something, and in this case they'll be involved in the transfer of a functional group from one molecule to another. Then we also have another class of enzyme that is known as the lysase enzyme. Yeah, that is lysase enzyme. So here it's an S, lysase enzyme. So basically these lysase enzyme, they are enzymes that are involved in the breaking down of chemical bonds. They are involved in the breaking down of chemical bonds. So what happens is that these lysase enzyme, they do that without the use of molecules like water or oxygen. They have got their specific molecules that helps them to carry out that function. Like for example, we have got cofactors. Those cofactors are one of the things that molecules like the lysase enzymes uses in order to carry out their function. So lysase enzyme, they are just involved in the breaking down of chemical bonds. These chemical bonds can be carbon to carbon bond, carbon to oxygen bond, or carbon to nitrogen bond, and also other covalent bond in a molecule. So they will just be involved in breaking down those bonds as they are trying to facilitate the formation of products. Then the fourth class of enzyme, it is hydrolase enzyme. Hydrolase enzyme or hydrolase enzyme. Hydrolase. So these hydrolase enzyme, these are enzymes that breaks chemical bonds using water. So they are also involved in the breaking of these chemical bonds, but the difference is that they use water molecules, and the process by which these hydrolase enzymes use water molecules to break the bonds is known as hydrolysis. So these enzymes are going to be involved in the catalysis or the breaking down of or the hydrolysis of bonds like the ester bonds, the peptide bonds, the acidic anhydride bonds, and also the carbon to carbon bonds, the ether bonds, and so on and so forth. So they are just involved in the breaking down of these chemical bonds using water, of which the process is known as hydrolysis, because it is the process by which an enzyme will actually use water to break down a bond. Then we also have the fifth type of classes of enzyme. And this fifth type is isomerase enzyme. So if we try to break down this name isomerase, we are going to find that it has got a name inside it, which is an isomer. Yeah. So what you have to understand here is that an isomer is basically a molecule that has got the same chemical formula with another, but they tend to differ in terms of atomic arrangement. So for example, what I'm talking about is that these isomerases, they are involved in the reactions that are involved in the changing of the structure of a molecule to form another molecule. So that structure of a molecule is going to be changed to form another molecule, but they will use the same atoms of that particular molecule, just rearranging them so that they can change the structure of that particular molecule. That is what these isomerase enzymes work on. So they are enzymes that catalyzes the structural change of a molecule. For example, let's say if you're having a molecule that is having, let's say, atoms that is STOP, which is basically, you can say, stop. Yeah. So when an isomerase enzyme comes and reacts on this molecule, maybe it is going to form a molecule that is going to have, maybe let's say, TOPS. So I'm going to find that these atoms that are in this molecule are going to be rearranged so that it can form a molecule with a different structure, but having the same atoms just arranged in a different way. So I'm going to find that it will form a molecule that will be having the arrangement of atoms like tops. But you know that these are the same atoms that were in the molecule, which is this one having a stop. Atoms in that arranged manner. So basically, isomerase, they just enzymes that catalyzes the structural change of a molecule. Then we also have enzymes that are classified as the ligase enzymes. Yeah, enzymes that are classified as the ligase enzyme. So I believe these terms are quite familiar, but we're just trying to add more flesh to the bone. So these enzymes, these ligase enzymes, these are just enzymes that catalyzes the joining of two molecules together. So they will just be catalyzing the joining of one molecule and another to form a new chemical bond to produce a new molecule. So they will just get this molecule and another molecule, they will just join them together by forming a chemical bond and produce a new product. That's just how these ligase enzymes, they operate. So one thing that you also have to know is that in as much as these enzymes are working like this and are being classified in that manner, there are certain non-protein molecules or substances that tends to help them work. So these molecules are also of different types. We have molecules like the cofactors, we also have molecules like the coenzymes, and we also have molecules like prosthetic groups. So let's start with the cofactors. So if you have a question, please feel free to ask and we'll be happy to attend to your question. So let's start with now cofactors. What are cofactors? So basically, these cofactors are non-protein chemical substances that help an enzyme to work properly. So what happens is that these cofactors, the cofactors are actually an enzyme without a cofactor, it is inactive. So when a cofactor binds to the enzyme will become active and it will be able to carry out its function. Remember, a cofactor is not the same as a substrate. These two are different. Thank you very much, Stella. Yeah, so these cofactors are actually molecules that are non-protein that tend to help the enzymes to work properly. So we are going to find that if an enzyme, let's say this is an enzyme that has got an active site and it is also going to have a site, and this site, it is going to be specifically for the binding of a cofactor. So let's say if this enzyme is not having a cofactor that has been bound at that particular site, this enzyme is going to be inactive. So just like the way I mentioned that these cofactors, they help in the activities of these enzymes Like they are molecules that helps the enzymes to become active, and they help these enzymes to carry out their catalytic functions. So one of them, we are saying they are cofactors. So these cofactors, they bind specifically, they've got their site where they bind. And when they bind there, they cause this enzyme to become active. So the cofactors, one thing that you have to take note of is that cofactors, they bind transiently. Cofactors, they bind transiently to an enzyme and reversibly. So this basically means that cofactors, they can bind and also unbind without changing the enzyme. So they can just go bind there, and they can also unbind from the enzyme, but they won't change anything about that, the structure or about that particular enzyme. Then by binding reversibly, that's the same thing that I'm talking about. They can bind and unbind from it. Then binding transiently, that means that they do not stay permanently attached to that particular enzyme. They can be taken off, or they are loosely, more like they are not attached permanently. They're just like loosely at any point they can unbind off, and they can bind back. So these cofactors, we have cofactors, for example, of cofactors like magnesium, also have iron and zinc. These are types of cofactors, and these specifically are called metal cofactors. And you're going to find that enzymes that require these metal cofactors for them to be activated are known as metal activated enzymes. That means that for that particular enzyme to be activated and to carry out its catalytic function, it requires a metal cofactor, which can be magnesium, zinc, or iron, for it to be activated. So that particular enzyme is then called a metal activated enzyme. That is what we have to know about cofactors. Then the other molecule that we have to know about is what we call a coenzyme. A coenzyme. So coenzyme, they are also small non-protein organic molecules that bind temporarily to an enzyme. So they bind to these coenzymes, they actually bind to enzymes, and they assist in the catalysis process of that particular enzyme. So I'm going to find that some coenzymes bind to these coenzymes, they have got a binding site that is known as coenzyme binding site, which is part of the active site. That's where they bind. So when they bind there, they are going to help in the catalysis process of that particular enzyme. So we have to know that an enzyme can be active or in an inactive state. So let's say, for example, we are having a diagram. This particular diagram is looking like this, or we're having an enzyme. Let me just say an enzyme. And this particular enzyme is looking like this. So this particular site here, it is the active site. It is the active site. And this particular site, it is the site for the binding of a cofactor. So the coenzyme, what will happen is that the active site will attract the binding of what we call a coenzyme, of which in these other terms we call a substrate. So we are going to find that this particular structure, we are going to find that at this point we have the binding of a cofactor. Then at this particular site we are going to have the binding of the coenzyme. Then this is going to be the enzyme. So now what happens is that the inactive form of an enzyme, or the inactive state of an enzyme, it is known as an apoenzyme. So it's more like just the protein part of an enzyme without its cofactors and without its coenzyme. It's just known as an apoenzyme. So I want you to know these terms. An apoenzyme, it is an inactive state of an enzyme. An apoenzyme, it means that one, a cofactor is not at its site where it binds, and two, and a coenzyme, it is also not at its site where it binds. So that protein part of the enzyme, which is this just part, that remains without the coenzyme and also without a cofactor, is what we call an apoenzyme, which is basically an inactive form of an enzyme, just the protein part of an enzyme which is not active. Then when the cofactor binds and its coenzyme binds and now starts facilitating the whole activity of an enzyme, helping it in its catalytic functions, that oath structure becomes what we call an halo enzymes or an holo enzymes. Yes, it becomes an holo enzyme. So we can simply say that the complete and active enzyme, it is known as an holo enzyme. So an holo enzyme consists of an apoenzyme, a coenzyme, and a cofactor. When these things they become together, the cofactor binds on its side, the coenzyme binds on its side, it will become active and it will be known as an holo enzyme. But if it is not active due to the cofactor missing or the coenzyme missing, both of them are missing at where they're supposed to bind, it is known as an apoenzyme, which is inactive state of that particular enzyme. So you have to know also how to draw the same structure and how to label it. It is very important because you might come across that question, and we'll show you the questions that falls under the same things. So the other things that you also have to know is what we call prosthetic group. Is what we call prosthetic groups. Prosthetic group. So prosthetic groups, they are also non-protein organic or inorganic cofactors. So these prosthetic groups, they are also non-protein inorganic factors that have got also the binding site on an enzyme. So what happens is that these prosthetic group, they bind tightly and permanently. And they bind tightly and permanently to an enzyme. So these, they have no mercy, like the way the cofactors are, like they bind transiently and reversibly. The prosthetic groups, they have no mercy, they just go and bind tightly and permanently. And they are very, very essential for the activity of an enzyme. So one thing that you have to know in case you come across a question that says, what is the difference between a cofactor and prosthetic group? One major difference that you should be able to remember is that prosthetic groups, they bind tightly and permanently to an enzyme, while cofactors, they bind transiently and reversibly. Okay, so now having known about these concepts, we can now try to look at what we call isoenzymes. So if you have a question right now, you can ask. Okay, so we are going to look at isoenzymes. So these isoenzymes basically are isoenzymes. So isoenzymes are just distinct enzyme forms that catalyzes the same reaction, or we can say these are just enzymes that are of different forms that catalyzes the same reaction. So they differ in terms of their regulatory mechanism and also the sensitivity as well as the substrate affinity. They tend to differ, these isoenzymes, and they often exist in different tissues. So we're going to find that there are some that are found in blood tissue, some that are found in muscle, some are found in the heart, some are found in the lungs. So they just differ in terms of where they are found. So they are distinct enzyme forms that catalyzes the same reaction, and they tend to differ in terms of their regulatory mechanism, sensitivity, and substrate affinity, as well as the sites where they are found. So one example, or an example that I can give under isoenzymes, is enzymes that are known as lactic dehydrogenase enzyme, or lactic dehydrogenase enzyme, that is abbreviated as LDH. So this LDH, it is a class of enzymes, or it is an example of enzymes that involves different types of enzymes under it. So we have got LDH1, LDH2, which is lactic dehydrogenase 1, lactic dehydrogenase 2 and 3, and up to somewhere 5. So these enzymes are actually very, very important because they are used in what we call diagnosis. They are used in diagnosis. So by that what I mean is that these enzymes, they are going to be used depending on the concentration of that particular enzyme in that particular body tissue. They can know to say, oh, for this enzyme to build up in that particular issue, it means that there is this that is happening with that tissue. That's why I'm saying that they are used in diagnosis or in the diagnostic processes that are used. So we have got the LDH1, lactic dehydrogenase 1. So these enzymes, they are used in the diagnosis of heart conditions, like let's say if someone is having myocardial infarction, you're going to find that LDH1 are going to be present. So they are going to be used in the diagnosis of heart conditions and also conditions that has to do with red blood cells, the LDH1. Then the LDH2, which is lactic dehydrogenase 2 enzymes, they are used in diagnosis of conditions that has to do with white blood cells. And also they can be found in small amounts in red blood cells and so on and so forth, but they are mostly in white blood cells, in conditions that has to do with white blood cells. Then you also have lactic dehydrogenase 3. These lactic dehydrogenase 3, these enzymes, they can be used in diagnosing conditions that has to do with the lung tissue. Yeah, so in lung tissue, you are going to find that these are going to be of help. And also lactic dehydrogenase enzyme 4, lactic dehydrogenase enzyme 4, they are going to be used in the diagnosis of condition that has to do with the white blood cells, the kidneys, the pancreas, and also the lymph nodes. Yeah, they are going to be used in such body tissues. So, and also we have the last one, which is lactic dehydrogenase 5. They are used in the diagnosis of conditions that has to do with the liver and the skeletal muscles. Yeah, so these enzymes are actually very, very important. So these are under isoenzymes. So because as we said, isoenzymes, these are distinct forms of enzyme that catalyzes the same reaction. Then they also tend to differ in their regulatory mechanisms, meaning that how they can be regulated, and also their sensitivity and the affinity to their substrate, they tend to differ, and they are found in different body tissues, just like the way we have seen here. So from there, we can now dig deep into what we call enzyme kinetics. We look at now what really happens when these enzymes are carrying out their functions. What really happens? So let's look at enzyme kinetics. Enzyme kinetics. So enzyme kinetic is actually just the study of how fast an enzyme works. How fast is this enzyme working? That is what has to do with enzyme kinetics. And it also has to do with how the enzymes attract the substrate to bind to the active site, and also what conditions tends to affect the rate of reaction for this particular enzyme. So that is what enzyme kinetics is all about. That's the study of how fast an enzyme will work and how they are going to bind their substrates on, and what conditions or how different conditions affect the rate of reactions that they perform. So I'm going to find that enzymes, they do not actually change the thermodynamics, but they tend to influence the kinetics, meaning that they just influence the rate of reaction for that particular enzyme. They don't bring about changing the energy that is involved between one molecule and the other, but they're just involved in influencing the rate of reaction. Just like the way we saw that they increase the rate of reaction by lowering the activation energy, which is the energy that is required for a reaction to take place. Yeah, so we know that the rate of reaction can be calculated by a simple formula that can be given as follows. We have a formula like V naught, which is the initial velocity or the rate of reaction of an enzyme, equals to the maximum velocity or the maximum rate of reaction for that particular enzyme with a given concentration of substrate over the Km plus the concentration of that particular substrate an enzyme is operating on. So the Vmax, as I have said, it is just the maximum rate of the reaction, and the Km is actually a constant. So this constant, the Km, it is a constant which is just it is the substrate concentration at which the rate of reaction is going to be half of the maximum velocity. So what I mean is that the Km, it measures how fast an enzyme is going to convert reactants into products. So in simple terms, I can say the amount of substrate that is required for an enzyme to work at half of its maximum velocity. So let's say if we are having a graph that is like this, and we know that the maximum velocity at which an enzyme can work, it is at this point, which is the Vmax. In half of the Vmax, we can put it at this point. Let's say this is half of the Vmax. So what happens here is that the Km will just be showing how much substrate is needed for an enzyme to work at half of its maximum velocity, or to reach half of its maximum velocity. So it will also tell you how strong an enzyme is attracting those substrate to bind to it, or how strong an enzyme is attracted to bind to its substrate so that it can reach half of its maximum velocity. So we are going to find that, before I show you the graph, one thing that you have to know the concept about the Km is that when the Km of an enzyme is low, it means that that particular enzyme, it is having high affinity. That particular enzyme is having high affinity. It is having high affinity to each substrate, or it is strongly attracting, or it is strongly attracted to its substrate. So meaning that the substrate will be strongly attracted, or they will be strongly attracted to an enzyme, so they will be going fast to bind to the active site of an enzyme. That is if the Km is low, meaning that the affinity for an enzyme to a substrate is going to be high. So meaning that the opposite is true. When the Km is high, it means that the affinity for an enzyme to each substrate is going to be low. So we have to master this concept, because it is going to really help us when we start looking at regulation or inhibition of enzymes, and also when we start looking at this graph. So we know that the Km, when is low, the affinity for an enzyme to each substrate is going to be high. When the Km is high, it means that an enzyme is going to have low affinity to its substrate, or it is going to have weak attraction. It won't be attracting each substrate that much. That is if the Km is high. So what you have to understand is, in a normal case, let's say in a normal case, and you have been brought, you do an experiment of an enzyme, or maybe it's a certain type of an enzyme that you are showing, and graphically it is presented that an enzyme reaction on a graph, it was going like this. So keeping in mind that this is the Vmax, meaning that is the maximum velocity, or that is the maximum rate at which an enzyme can speed up that reaction. This is the Vmax that we are having here. So we said that the Km will just show how much of the concentration of substrate that is required for an enzyme to work to reach at half of its maximum velocity. So the half of maximum velocity is here. So meaning that the Km in this particular reaction of an enzyme, it is going to be noted by this line, and you are going to indicate here that this is the Km. So meaning that in this reaction, the substrate, the enzyme only required this amount of substrate from this point to this point in order to work to reach at its half of its maximal rate of reaction, or half of its Vmax. Then the other thing that you have to know is that, let's say for example something happens and an inhibitor comes in. You're going to find that the Km, the Vmax, these things are going to be affected, because that inhibitor is somehow either reducing the activity of an enzyme or lowering its energy to perform the same reaction, and so on and so forth. So we are going to look at how the inhibitors they tend to affect. But one thing that you have to understand here is that the Km basically just tells how strong an enzyme is going to be attracted to each substrate, or the amount of substrate that is required for an enzyme to work at half of its maximum velocity. How much substrate do we need so that an enzyme can work fast and reach half of its Vmax? That is what the Km is just going to be showing. That's why we are saying that when the Km is low, it means that an enzyme is having high affinity to each substrate. So that shows that it is going to work faster and reach half of its Vmax within a short period of time. But when the Km is high, it means that the enzyme is having low affinity, so it is struggling into attracting the substrate to bind onto its active site. So you're going to find that its Vmax to be reached, to even reach half of it, it is going to take time. So if you have questions, please ask. Okay, so let's now look at factors that will now be affecting the rate of reaction for the same enzyme. So you know that enzymes can be affected by inhibitors, and these inhibitors, we have what we call competitive inhibitor, non-competitive inhibitor, and also uncompetitive inhibitors. So let's now try to look at a condition in which you are having a competitive inhibitor present. So what happens? So we know, first of all, what you have to know is that a competitive inhibitor, these are just molecules that binds to the same site as the substrate. So in short, they are going to be competing with a substrate to bind to the active site. It's more like you are having one parking spot for a car. Then there are two cars. You are in your car and also your friend is in a car. Then you see that, oh, we are only having one parking spot. So whoever gets there will park there. So you speed to make sure that you get there fast than your friend, and also your friend will have the same thing in mind. That's what happens. So a competitive inhibitor will just be competing to bind to the active site. So it will be competing with the substrate. So what happens is that this competitive inhibitor, if it succeeds and binds to the active site, it is not going to be worked on by the enzyme and then it is going to be formed into products. No, it does not. It is not changed by the enzyme. No. And one thing that we can do in order to compensate this, to say, maybe let's say you have seen that you have a competitive inhibitor in a certain reaction, you can just try to increase the substrate concentration, because you know that when you are having more substrate that are involved in that reaction compared to the inhibitors, you have a high chance of a substrate binding to the active site compared to an inhibitor. So we can just simply say that competitive inhibitors can be overcome by increasing substrate concentration. So now let's look at how now this competitive inhibitor tends to affect the Km, the Vmax, and the half of the Vmax, and how does it affect that? So what you have to know is that in the presence of a competitive inhibitor, let's say this is an enzyme, and this enzyme finds that instead of having its substrate binding to the active sites that we are having here, these are the active sites, you're going to find that maybe an inhibitor comes and binds there. So this inhibitor that binds there, how is it going to affect the rate of this enzyme, and what is going to happen to the Vmax and the Km? We know that the Km will just show how much of this substrate are needed so that an enzyme can reach half of its Vmax. So now when a competitive inhibitor binds to the active site of this enzyme, what will happen is that in a scenario of a competitive inhibitor, the Vmax is going to remain the same. This maximum velocity or the maximum rate of reaction is just going to remain the same. It's not going to be altered. It is not going to be changed. What will be changed, as well as the half of the Vmax, is just going to remain the same. But what is going to be changed is the Km. So the Km is going to be increased, and we know that when the Km increases, it becomes high, the affinity for that enzyme to each substrate is going to be low. So you're going to find that most enzymes in a reaction, let's say having a culture media, or let's say an environment where you are putting these enzymes and you are trying to see how they are going to work on the substrate to produce this product, and in that same culture media you have put competitive inhibitor, you're going to find that the rate of reaction for that particular enzyme, for the rate of reaction at which those products are going to be formed, is going to decrease. Why? It's because the competitive inhibitor have already bound to the active site of this enzyme. So meaning that they have increased the the Km for this enzyme, meaning that you require to put in a lot of substrates for you to make sure that the chemical reaction is done at its normal time. Just like the way we said that competitive inhibitor can be overcome by increasing the concentration of the substrates. So what happens is that in a competitive inhibitor, the Km is going to increase. So instead of it being here at its normal position, the presence of a competitive inhibitor, a graph will start looking like this. So a graph will look like this, and then it will definitely reach the Vmax, but the rate at which it is going to reach at the Vmax is going to be different in a normal condition where a non-competitive, or rather a competitive inhibitor is present. So this shows that now the Km is going to be increased. So one thing that you have to master about this competitive inhibitor is that the Km increases, but the Vmax remains the same, and the graph is going to be looking like this. So it is going to be appearing like this, in which you are going to see the normal graph where there is no inhibitor, and here you are going to have the presence of an inhibitor, and specifically this is going to be a competitive inhibitor. So this is what happens. Then we also have another one that we call a non-competitive inhibitor. So basically a non-competitive inhibitor is just an inhibitor. For it does not compete with the substrate to bind to the active site. No. For it binds to each specific site that is none other than the active site, which is the allosteric site rather. So for the non-competitive inhibitor, it binds to the allosteric site of an enzyme. It is going to bind to the allosteric site of an enzyme. That is for a non-competitive inhibitor. So they are not going to compete with the substrate to bind to the active site. And the substrate can still bind to the active site of an enzyme. By the fact that a non-competitive inhibitor is bound to the allosteric site of an enzyme, what will happen is that it is going to reduce the catalytic power of this particular enzyme. So we are going to find that if this is the enzyme that we are having, we are having an enzyme which is this one here, and then a non-competitive inhibitor comes and binds to the allosteric site of an enzyme. The substrate can still come and also it can bind to this, or the substrate can come and bind to the active site of an enzyme. But what will happen is that when this non-competitive inhibitor binds to the allosteric site, it is going to reduce the catalytic power of an enzyme. So meaning that this enzyme, the energy that it had, that motivation to convert this substrate into products is going to be reduced. So you're going to find that this is because the active site will become less sensitive due to the presence of a non-competitive inhibitor binding there. It's more like you're having a chair, and that chair, it is having a broken, it is broken on each one of the four stands. You're going to find that even if you sit there, you're not going to sit stably, because that chair is not having the complete four stand that it's supposed to have. So you're not going to sit as you are supposed to be sitting in a four proper stand chair. So this is what happens. So when a non-competitive inhibitor binds to the active site, it is going to make the enzyme become weak. It will reduce the catalytic power of that particular enzyme. That is because it will make the active site to become less effective for substrate, so that it can convert the substrate into products. So what this non-competitive inhibitor does is that for the Vmax is what it is interested in. So I'm going to find that the Vmax of this particular enzyme is going to decrease. Instead of reaching its maximum Vmax like the way we saw, it is going to decrease. The Vmax is going to be decreased. It will fail to reach there because its catalytic power has been reduced. But the Km is just going to remain constant. Meaning that the amount of substrate that is required to bind to the active site of an enzyme so that it can work half of its Vmax, they're just going to be there. Or they're just going to be the same amount. But the power for now this enzyme to convert this substrate into product is what is going to be affected. It is what is going to be reduced. So as a result, it won't be able to reach the Vmax that it was supposed to reach. For example, this is the Vmax that we're having here. Meaning that this enzyme won't be able to reach there, but the Vmax will drop. So if the Vmax will drop, even half of this Vmax, if half of this Vmax is also going to drop. is also going to drop. Let me use this diagram here as we have seen. So we are going to find that this is a graph that will be showing a normal reaction of an enzyme where there is no inhibitor, and in this case is a non-competitive inhibitor. But if you find that the Vmax was there for a normal reaction where there was no inhibitor, and there is a presence now of a non-competitive inhibitor and binds to the allosteric site, this enzyme will fail to reach the Vmax because its catalytic power has been reduced. So what will happen is that the Vmax is going to reduce. So if this Vmax reduces to this point, it means that even its half Vmax is going to reduce. So what will happen is that the Vmax will reduce, and this half of the Vmax will reduce, but the Km is going to remain the same. The concentration or the amount of substrate that is required for this enzyme to work at half of its Vmax is just going to be the same. That's why you are seeing in this graph the Km is just constant, but the Vmax is reduced rather. So that's one thing that you have to keep in mind to say. The Vmax reduces when a competitive inhibitor is present, and the Km remains constant. But for that is for a non-competitive inhibitor, but for a competitive inhibitor, we are going to find that the Vmax will remain the same and the Km will increase, meaning that the Km will shift this side to increase, just like the way we saw in the previous graph. I believe we are following. If you have questions, please feel free to ask. And we also have another type of inhibitor which is overshadowed mostly. It is not talked about, but for the sake of knowing, let's try to dig deep in and know it. So we have also an inhibitor that is known as a competitive, or a uncompetitive rather, not competitive or non-competitive, but uncompetitive. Uncompetitive inhibitor. So this uncompetitive inhibitor, basically, it also binds to the allosteric site of an enzyme, but it does not bind to an enzyme, but it binds to the enzyme substrate complex, meaning that it is going to bind to an enzyme which is already having its substrate. So when a substrate and an enzyme have already bound together, they form what we call enzyme substrate complex, because it is a complex that is formed of an enzyme and its substrate. So it is just going to be interested in that one. So it will go and bind to an enzyme substrate complex. So the purpose of it going to bind there, it is to make sure that that enzyme substrate complex, it goes and shuts down the enzyme so that the enzyme won't be able to produce the product. So it will prevent the enzyme substrate complex from forming products, and by that it is going to inhibit the same complex from working. So it is going to shut it down. And this one, it cannot be overcome by adding more substrate because it is shutting the whole thing down. And what happens is that in condition of the kinetics, the enzyme kinetics, the Km, when a competitive inhibitor is present, the Km is going to decrease, and also the Vmax is going to decrease. This is because when an enzyme complex, enzyme substrate complex is shut down, you are going to find that the Vmax is going to decrease. Why? Because that enzyme won't be able to reach the Vmax because it is already more like it has been, the energy out of on it has been sucked, such that that enzyme is now inactivated, and the Km also is going to decrease. Yeah. So having talked about that, we can now look at enzyme regulation. So the same enzyme that we have been talking about, how are they regulated? What are the mechanisms that are involved in the regulation of these enzymes? At this moment, you are very free to ask a question before we dig deep into enzyme regulation. Okay, so enzyme regulation. So we have got the mechanisms that are involved in enzyme regulation. Yeah, so we have enzyme regulation. That's what we are going to talk about. So enzyme regulation, basically, we have got 4 types of enzyme regulation, and you have to pay attention to this. They are also very much asked about in essay questions, also in MCQs. You are going to find them. We'll show you the questions how they are phrased under enzymology. So enzyme regulation, we have got four types in which four ways in which enzymes are regulated. So let's just write enzyme regulation. So we have what we call allosteric regulation. We also have what we call covalent regulation. Then we also have what we call feedback inhibition. Then we also have the last one that we call genetic. We have what we call genetic regulation or gene regulation of an enzyme, genetic or gene regulation. Okay, so now let's try to look at the regulation of these enzymes. So let's start with this one, the gene regulation. So we know that enzymes, just like the way we talked about, most of the enzymes are protein in nature. So meaning that there is that process in the background, that is the process of transcription, translation, and modification to form a particular protein, and that protein will now become an enzyme, let's say for instance in this condition. So there are specific genes that codes for amino acids that are involved in the formation of proteins that are just used to form an enzyme. So under genetic regulation or gene regulation, you're going to find that it will refer it actually a type of regulation in which there is a control of enzyme synthesis at the gene level. So it determines how much of these enzymes are going to be produced and when they should be produced, because we know that the body has got that mechanism or that system of making things when they are needed, and if they are not needed, it is just going to shut down those genes. It's just going to switch them off. And if the particular genes are needed to produce certain proteins that are needed in the body at that particular time, those genes are going to be switched on. That is what happens under genetic level for the regulation of these enzymes. So you're going to find that there are certain there are certain genes that codes for the same amino acids that are involved in the formation of proteins that are required to form enzymes, specific enzymes. So those genes, when we see that a specific enzyme is not needed, they are going to be switched off. And when that particular enzyme is needed, they are going to be switched on so that that enzyme can be produced and can be used by the body. So they are just going to be regulated in that. So when they are required, the regulation will be positive, meaning that there are going to be formation of that particular protein to produce the enzyme that is needed. If they are not needed, the regulation will be negative, meaning that those particular genes are going to be switched off so that the enzymes that is working there can stop working because the body does not need it anymore. That is genetic regulation. Then when we talk about feedback inhibition, this is where now the end product of that particular reaction or pathway goes back and inhibits the enzyme that is involved in the pathway. So let's say, for example, this is a pathway. So in this pathway, there is the formation of this molecule. From this molecule, you're going to form this molecule. Then it goes like that. So let's say at this point here, we are having an enzyme that is operating. So this enzyme is